Genome sequencing of Icelandic population. Introduction of Icelandic population. Iceland is a Nordic island nation located in the North Atlantic Ocean. The population of Icelandic are around more than 360,000, and 91.1% of the residents of Iceland are Icelandic citizens. They study human genome and its impact on disease and other traits, focusing on the Icelandic population due from genealogy of the nation and the amount resident of the nation. The methodology used for the study is 1. By withdrawing blood samples of participants. 2. Collect information of the family history of participants donating blood into the study by including the phenotype of first and second-degree relatives and interrogating over their possible genotype and phenotype. For the result and the discussion of the study, from the whole genome sequencing of Icelandic participants, the reading of the sequence shows that variants of mutation can be read from the database. The second variant of mutation contains 4 categories. 1. Loss of function, including stop gain or loss variants, frameshift indels, splice donor or acceptor variants, and initiator codon variants. 2nd. Moderate impact, including missense variants, in-frame indels, and splice region variants. 3rd. Low impact, including synonymous variants and 3' and 5' UTR variants. Lastly, deep intronic and intergenic variants. The percentage of variants in the sequence of Icelandic population shows that low impact is 21%, moderate impact shows 25%, loss of function is 34%, and other categories is 20%. Type of disease from the variant sequence. 1. Mendelian disease, due to conservative variants, loss of function, and moderate impact. 2nd. Atrial fibrillation, most common sustained cardiac arrhythmia. Gene MYL4 undergoes frameshift deletion, cause an early onset of atrial fibrillation. 3rd. Liver disease. Gene ABCB4 become mutated from GWAS of gallstone disease lead to liver disease. Variants that happen on gene ABCB4 are missense SNP encoding P-glycine 622 glutamic acid, frameshift insertion encoding P-leucine 445 glycine in ABCB4. BVVL syndrome. BVVL syndrome is a Brown-Violletto von Lery syndrome, a rare degenerative disorder of neurologic condition caused from a single missense mutation in the riboflavin transport gene SLC52A2. The discussion from the study by sequence the whole genome of large group of Icelandic provide 1. A comprehensive understanding discover association between variants in sequence and phenotype. 2nd. The density and frequency distribution of sequence allow to evaluate sequence engine and notation. 3rd. These fewer very rare variants in a small isolated population have found than in larger population. However, this small-sized population allow deleterious variants to higher frequencies.